In this video, we're going to be going over the details of the nervous tunic, or retina, which is the inner layer. So remember, we had our fibrous layer out here, which is the sclera and then the cornea. Then you had your vascular layer in through here, or tunic, which consists of the choroid, your ciliary body processes, and iris. And the innermost layer is called your retina. And the retina is going to have neurons involved with photoreception, the rods, the cones, and then those neurons are going to help to transmit those impulses into the optic nerve. The retina consists of two layers. You have a pigment layer, pigment epithelial layer, which is here, and this is at the base right next to the choroid. Then everything distant out from that, that's basically going into the inner portion of the eyeball, is going to be your nervous layer. So the pigment layer lies between the choroid, as shown here. Remember, the choroid is in blue in this diagram and wraps all the way around. The retina is just inside of that. And then right next to the choroid is this layer of the retina called the pigment epithelium. It also consists of melanocytes that are producing melanin, and these are going to help to absorb stray light along with the choroid. Shown in this image is an albino, and you can see the red iris because of the lack of blood vessels. Well, also, if you look deep inside the eye through the pupil, it's also going to look red in here because it doesn't have the melanin, doesn't have that pigment, both in the pigment epithelium and in the choroid layer. Well, in addition to synthesizing melanin to absorb stray light, that pigment epithelium also functions to phagocytose the shed outer segments of the photoreceptors. Shown here, the photoreceptors, I'm going to see that these photoreceptors have these discs that have photoreceptors in these, and these discs get continually produced here at the cell body of the photoreceptor, and then those discs are going to be pushed out into this pigment epithelium. Well, the pigment epithelium is going to degrade or phagocytose these old discs as new ones are formed here near the cell body. The older ones are going to be phagocytosed here, and they phagocytose about 10% of these discs per day. The pigment epithelium also delivers nutrients and blood to these photoreceptors. And here's an actual slide showing the pigment epithelium with the choroid, and then you have your photoreceptors here, and various other cells such as the bipolar cells and the ganglion cells. They're going to help transmit impulses from those photoreceptors to what's going to become the optic nerve. So the photoreceptors, including rods and cones, when you look at your rods, you have about 120 million per retina. They're specialized for vision in dim light, such as moonlight. There's no color vision seen with these rods. It's only the black and white vision, or night vision. The cones, you have a lot fewer cones in the eye. Each eye has about 6 million cones. They're specialized for color vision, and they have a cone shape versus the rod shape of the rods. Now, they contain 3 types of cones, and these are blue cones, green cones, and red cones. Now, it's not referring to the actual color of the cone, it's just referring to the color that the cones are sensitive to. So blue cones are sensitive to blue light, green cones to green light, red cones to red light. And color vision results in a various combination of these 3 types of cones. Now, the cones are also specialized for the sharpness of vision. In fact, we're going to see at the focal point you have only cones in that area. If you have, for example, a defect to where you're not producing the right pigments for the red and green cones, then you can have what's called red-green color blindness. It's a sex-linked genetic trait. It's found on the X chromosome. So if you have a defect on this, on your X chromosome, and you're a male, then you're going to have red-green colorblind. For a female, it'll require 2 defective genes on each X chromosome. So you see a lot more males with red-green color blindness than you have females. If you have normal color vision, you should be able to see this 7 in here. You would see in this picture a woman with a red dress against a green background. Here she is with the red dress against a green background. Somebody who has red-green color blindness, they have a tough time telling between red and green. So here's the same dress of somebody who have red-green color blindness. You're looking up here at the bulbs. Here's a green, red. Here's what they look like here. You'd see different shades of them, but they look basically very similar in color. Here's fruitcake. Somebody who's red-green colorblind would see this. And red suit, green bag, this is what a red-green colorblind person would see. For blue, it looks the same. There's no difference in the blue color. Well, if you look at the structure of rods and cones, these are specialized neurons that consist of an outer and an inner segment. The outer segment contains visual pigments embedded in the membrane discs. Shown here, remember there's a pigment epithelium, these are the cones, and they're cone-shaped. And you have your rods, which are more rod-shaped, and they have these discs here that are embedded in the pigment epithelium. In these discs, shown along here, we're going to find that there are photopigments that will help to detect and get stimulated by various wavelengths of light. These discs have a phospholipid bilayer with a photopigment embedded in that phospholipid bilayer. A portion of the outer segment is what is embedded in the pigment epithelium. Now the inner segment of this, shown here, consists of the nucleus and major organelles that help synthesize those visual pigments. They also have at the very end the synaptic endings here that are going to synapse with the bipolar cells. So in looking at this here are the bipolar cells, and they're going to synapse with these synaptic endings of the rods and cones. Those bipolar cells will send information to the ganglion cells, and then those ganglion cells, the axons of which are going to become the optic nerve. So when you look at the visual pigments that are embedded in these discs, they consist of two components. They have opsin and they have retinal. Looking at this image here, this is one of the discs, let's say from a rod or a cone, and in this disc you're going to find these opsin proteins. These are integral membrane proteins, which means they go all the way through this phospholipid bilayer that you can find here in the disc. Within each of these opsins, you're going to find a retinal pigment. This is a light-absorbing part, and it's synthesized actually from vitamin A. So vitamin A is important for vision because, well, vitamin A is used to synthesize retinal. This is what's going to change shape in response to various wavelengths of light that can come through the opsin. So rods and cones contain the same retinal pigment, but differ in their types of opsin that hold that retinal. Rods are going to contain rhodopsin, and cones will have derivatives of this rhodopsin, which are just called the color pigments, or photoopsins. Now the type of opsin is going to determine the wavelength of light that can be absorbed by the retinal. And looking at this, for example, you have blue cones, green cones, and red cones. Well, the cones themselves aren't colored, it's just the wavelength of light that they happen to absorb. And what determines the wavelength of light that they absorb is going to be the type of opsin that you have. When that wavelength of light gets absorbed, it's going to change the shape of these retinal molecules in here. Their stimulation in various combinations is the basis for color vision. Now the new discs containing visual pigments are continuously assembled at the base of the outer segment. So shown here is this outer segment, and here are those discs, and they're continuously produced. As new ones get made, it pushes the old ones closer and closer towards the pigment epithelium. After about 10 days, the discs are shed at the outer segment tip and phagocytosed by the pigment cells. So it takes about 10 days for these discs to move all the way to the pigment epithelium. Shown here are the discs, and here's the opsin, here's the retinal, and we're going to see that when light stimulates the retinal, it goes from a cis conformation, which is more of a C-shaped conformation, to a trans conformation, which extends straight out. So in photoreception, we have what are referred to as the light and dark reactions. So shown here is the photoreceptors in the dark, which release these inhibitory neurotransmitters that stop the bipolar cells from activating the ganglion cells, which send a neural impulse to the brain via the optic nerve. When light comes in, we're going to see that these rods or cones stop releasing inhibitory factors. And without the inhibitory factors, the bipolar cells can then stimulate the ganglion cells to send an action potential to the brain to indicate what you're seeing. The details of this, if you would like to discuss them, I'll be happy to go over the light and dark reaction and how actual neural transmission is being sent via that ganglion cell. However, this portion right here I am not going to test on, and I'm not going to go through the details in this lecture. You can read through these are in your notes, but I'm not going to go through these details, and you won't be tested on the light and dark reaction. So this portion right here in your notes, go ahead and put FYI. The only thing that you need to know is that you have this retinal pigment that sits inside of that opsin, and when retinal gets activated by the wavelength of light that comes through that's determined by the opsin, it's going to go from a cis conformation or shape to a trans conformation. So go from 11 cis conformation to its more active 11 trans conformation, and that signal, that photoreceptor, will send an impulse to the brain that this is what you're seeing, or these visual pigments have been activated. Recovery after stimulation, put FYI. You can read through here and ask questions if you like, but I'm not going to test you on this. This actually you need to know. It's a clinical application for night blindness, and individuals who have a deficiency in vitamin A, this can result in a decline in the retinal pigment and the corresponding decrease in vision. At first, daylight vision is not affected because of the increased amount of photon stimulation activates the remaining visual pigments. So those photons are what come in and activate those photopigments. However, if you have a decrease in vitamin A, you're not going to make as much retinal, because remember, retinal is derived from vitamin A. So vitamin A, which you find in carrots and beets and various vegetables, the vitamin A is used to make retinal. And if you don't have enough vitamin A, then you can experience night blindness. Basically, this problem first becomes apparent at night, which dim light proves to be insufficient to activate the rods and can be treated by just administering vitamin A. Dark adaptation, you can put FYI.